Now, the main cell of the nervous system is a neuron, and neurons can get pretty complicated. And if you're trying to learn about neurons and you're flipping all over pages to get information and sort it out and put it all together, that can be tough. So let's get that information right here on one piece of paper. First, let's organize our paper. I'm going to fold it in half long ways with a hot dog fold like that. And then I'm going to fold it into thirds the other way. So let's fold that over. And those should be about equal. And then we'll fold this side over the top and then unfold it. Now our paper is divided up into 6 boxes to help keep us organized. So this is all about how neurons work. We'll use this box here for neuron anatomy. Down here in these 2 boxes, we're going to talk about something called an action potential. Action potentials are the electrical signals of neurons. Now, action potentials can actually be modified to be really fast, and that's what we're going to talk about up here. And we'll use this box for something called saltatory conduction. That's for this box, and then we'll use these last 2 boxes over here to talk about the chemical signaling of neurons, which is called synaptic transmission. So let's get started by drawing a neuron. What do they look like in the first place? So a neuron has 3 basic parts, and I'm going to do my best to draw those 3 basic parts right here. So it's got this part with a nucleus right there, and this part of a neuron is called the cell body, and sometimes it's called the soma. Now, the soma has this cool shape because there's some other stuff going on here, so I'm going to draw some branches coming off of there. And this area of branches is called the dendrites. Dendrites. The job of the dendrites is to receive input signals. And that input is sent to the cell body. Now, a neuron will probably have to send a signal to another cell, and it uses a really long process to do that. I mean cell process, not like time-wise. The time that a neuron sends a signal is actually very quick. And that long process ends in, we're going to draw it like this. And we'll draw another one of those things kind of going out this way. And so this long part altogether is called, let's draw a little label like this. This whole thing is called an axon, kind of like the axle of a car where it's really long. The axon is the output area of. The dendrites receive the input, the axons send the output. Now, one thing that I want to make sure that we are clear on here at the very beginning, this is a neuron. So a neuron, where do we want to write this? I'm going to write it right here, really small. So a neuron is a cell, and a nerve is a bundle of neurons. All right, so we just want to make sure that we're being professionals with our terminology there. Now, in axon, I drew some special shapes here. This part of an axon where it's the thickest is called an Axon Hylic, and that's going to be important in our story a little bit later on. And then these little parts at the end of the axons are called axon terminals. Right? And terminal means end. So that's like these parts right here. Axon terminal. And those are really specialized to help with signaling as well. All right, so I told you that a neuron has three basic parts, and those parts are the dendrites, the cell body, and the axon has a little bit extra of specializations. The dendrites have some specializations too, with all of these branched structures that look like tree branches. Now, we're going to go over to this section to talk about action potentials. An action potential is the electric signal sent by a neuron. And an action potential happens on an axon. So what we're going to do is we're going to take a small section of axon and we're going to blow it up. We're going to blow it up over here. So let's draw that in here. We'll give ourselves a little bit of space and go about halfway down our box. So this is an axon, and over here is outside of the cell, and this is outside of the cell, and this would be the space inside. Now, in order for a nerve to send an electric signal, or a neuron to send an electric signal, a neuron uses charged atoms. And hopefully you remember from your chemistry class that charged atoms are called ions. Now, the ions that we're going to be keeping track of are going to be two different ones. One is going to be sodium ions. So when we're talking about sodium ions, I'm going to use this color. And the other is going to be potassium ions. And when we're talking about potassium ions, I'm going to use that color. Now, there's actually an imbalance of sodium and potassium ions, and that's critical for how a neuron works and signals. So how that imbalance is created in the first place is to use a specialized protein that's in the membrane. And I'm going to go ahead and draw this. And color it in. So this part is the part that has to do with potassium, and there's two little grooves in there. And this is going to be the part that has to do with sodium. And I drew three little grooves in there. This protein right here is called the sodium potassium pump. And the job of the sodium potassium pump is to pump 3 sodiums out and 2 potassiums in. And what ends up happening is we get a lot of sodium ions out here on the outside that build up, and a lot of potassium ions on the inside of the neuron. And now we've built up this difference. And you can see if we're putting more positive charges on the outside than on the inside, what ends up happening is the outside is more positive than the inside, or you can say that the inside is negative relative to the outside. Now, I want to draw some of these, since this is the outside of the cell too. I'm gonna draw some more sodiums out here, because sodium is high on the outside of the cell. Now, down here, I'm going to draw a little graph. So let's set that up. And this will all come together, I promise. So there's our graph axis. And on the x-axis, we're going to put time. And a signal of a neuron happens in just a few milliseconds. And on our y-axis, over here, we're going to put voltage. And we're talking about millivolts. So, you know, yeah, we're talking about voltage and electricity here, but not like huge amounts. Now, a voltage that we want to keep track of is minus 70 millivolts. And I told you that here, because there's only 2 potassiums for 3 sodiums, the outside is more positive and the inside of the neuron is more negative. And that amount of negativity on the inside is 70 millivolts. So right here, minus 70 millivolts. And we call this part the resting membrane potential. Resting membrane potential. And I'm going to put a 1 there because that's our first phase that we have to keep track of. And I'm going to write a 1 up here because that's what we're talking about. Now, during rest, what happens is sodium is getting moved out and potassium is getting moved in. Now, a good mnemonic to remember this is to think about this, a neuron at rest, as a salty, because salt is sodium chloride, right? A salty banana. And it's a little bit goofy, but what you're imagining here is a banana, that bananas are known to have high potassium, a banana rolled in salt. That is sodium chloride, salty banana. All right, so that's our neuron at rest. Then what happens when a stimulus comes that causes a neuron to signal? There's going to be a big change in the membrane potential, and it's going to spike like this. And this is phase 2 of our action potential signal, and it's called depolarization. And what you see is that the voltage of the membrane goes up. And it reaches a peak at about plus 40 millivolts. So if you think about the distribution of these ions, how could we make the inside become more positive? Well, we can't get any more sodiums or potassiums out, sodiums out. We can't get more potassiums in because that's already distributed like that. And so what we need to do is we need to bring some of these sodiums back in. And so there's another protein here, and I'm just going to draw it as a simple rectangle. And this is called a sodium channel. And how sodium channels work is at the right time, they open up and these sodium ions rush through the sodium channel. And go back into the neuron. And so now at this point, we've got a mixture in there of sodium and potassium. And we still don't have any potassium on the outside, or, not a whole bunch. So we've brought a lot of positive charges on the inside. So this is segment 2. Segment 3 of this signal, so this is an electricity change. Segment 3 of this signal involves the redistribution of potassium ions. And that's called repolarization. All right, so this is section 2. Section 3 is over here. And you can see that now the voltage is going back down. Well, we've got a whole bunch of positive ions on the inside now. And we can make the voltage on the inside go down if we use a potassium channel. So this guy is a membrane protein called a potassium channel. And what the potassium channel does is it will take some of these potassiums and shoot those potassiums to the outside. So we're losing positive charges on the inside, and that's why our membrane potential is going down. And still there's a lot of sodium on the inside. And then we've got our next phase here, and what actually happens is there is an overshoot of the minus 70 millivolts. And then the cell has to recover a little bit. So this part right here, phase 4, is called hyperpolarization. And hyperpolarization is a recovery period. Let me get another color here. So that recovery period down here is called the refractory period. And that's a time when a neuron can't fire another signal because it's trying to reset things up. All right, so phase 4 over here, now we've got everything flipped. So now we've got high sodium on the inside, high potassium on the outside, and we need to flip that back. And so during hyperpolarization and setting things back up to resting potential, we rely on our old friend, the sodium-potassium pump. So 2 grooves in there because we're pumping 2 potassiums in. And 3 grooves on the sodium part because we're pumping 3 sodiums out to get that salty banana again. So an action potential sends a wave of electricity down the axon. That electricity is caused by ions that are set up one way to start, and then they flip their distribution in a wave. First, the unequal distribution is set up by making a salty banana where sodium is pumped out and potassium is pumped in. When there's an appropriate stimulus, sodium rushes back in. That's the big electrical surge. Then that triggers for potassium to go out, and now everything is completely flipped, so that the whole process needs to be reset here so that a neuron can fire again. And that happens in a matter of milliseconds. So that can be a little bit confusing, and what I would recommend is just kind of getting a little bit of practice. Now that you have this, go to your book and read a little bit about it, and then come back to this and maybe look at another source until you've kind of made sense of things. Now, even though this happens in a matter of milliseconds, action potentials can happen even faster using a technique called saltatory conduction. Now, if you speak Spanish, you might know the word saltar, which means to leap, and saltatory conduction comes from the same word root as saltar. So let's go ahead, just for good practice here, let's redraw our neuron. So here's our neuron, and we've got our three main parts. We've got our cell body right here, and I'm going to draw a cute little nucleus in there. And then we've got these branched dendrites coming off to receive signals. And we've got our long axon, and then the axon terminal buttons over here. Now, a neuron like this is called non-myelinated. And this action potential has to happen along the whole length of the axon. But there can also be myelinated neurons. Myelinated. So let me draw that again. And what's different here, well, we've got the same basic parts. You can see the dendrites that I'm drawing in right now, and the cell body and the axon. But what's different is that there are these helper cells, and these helper cells cover parts of the axon. So I'm going to draw one, I'm going to draw a second one, third one, and a fourth one. So these purple guys right here are specialized types of cells, and they're called Schwann cells. And Schwann cells are specialized because they make myelin. This myelin material from the Schwann cells helps to insulate the electrical signal. So just like wires that you have metal in the middle and a plastic insulating coating on the outside, that's how this is working too. Now, the inside parts here, in between the Schwann cells, they have a special name too. And those are called nodes. And they have a special name, nodes of Ranvier. And Schwann and Ranvier are some people that kind of figured out what's going on here. And what's different about here, what's different in a myelinated axon compared to a non-myelinated, is the non-myelinated needs to send the signal all the way down. But in a myelinated axon, the signal jumps, the signal leaps from one node to the next. In other words, the only part of the axon that need to do this sodium-potassium pumping, then bringing sodium back in, and then putting potassium back out, the only parts that need to do that are the nodes. And as a result, myelinated axons conduct action potentials really, really fast compared to non-myelinated axons. Myelinated axons, they make up the parts that are called white matter in the brain and spinal cord. And when you need a signal to go really fast, you use a myelinated axon. Now, we've got one last part on here, and that part is synaptic transmission. Now, I drew these axon terminals over here. So when an action potential gets to the end of the axon at those axon terminals, then what? Well, there's usually these axons, and a neuron is attached to another neuron. So we can draw another neuron in here. That looks really poor. I'm sorry about that. Let me try that again over here. All right, so that's my really quick neuron right there. And so the synaptic transmission happens at the connection of one neuron and the next one. So an action potential gets to the end, and that signal needs to be sent to the next neuron in the chain. And so what I want to do is now I want to zoom in on one of these interactions right here. So let's go ahead and zoom in on that. And here is one cell, and this is going to be our axon terminal. And this cell is called the presynaptic cell. And the part that we're especially zooming in on, this is the axon terminal of that cell. And this neuron is going to talk to the next neuron in the chain. So let's draw the next neuron in the chain. So if this one is the presynaptic cell, you can probably guess that this guy is called the postsynaptic cell. Now, this setup right here of one neuron, the gap in the next neuron, this setup is the synapse. That whole thing. And a lot of people think of just this space as the synapse, but this part is actually just called the synaptic cleft, is the gap. Now, I want to talk about some specializations here. Now, the presynaptic cell has the specializations of some vesicles in there. I'll draw 4 of them, but there's a whole bunch. And this guy is called a synaptic vesicle. We know from our studies before that a vesicle is involved in storage and moving stuff throughout the cell. So what are we trying to store or move? Well, that's the contents of this vesicle right here. Draw that in green. I'm gonna use a lighter green. Let's see if this green shows up better. And these green particles that I drew are neurotransmitters. So the neurotransmitters are in the presynaptic cell, And this is the chemical message that's sent to the postsynaptic cell. So the ions in our action potential are the electric message. The neurotransmitter is the chemical message. And the postsynaptic cell can receive this message because it has receptors on its surface. Now, we talked about the area where neurons receive input. So this is going to be the dendrite of the next neuron in the chain. And let's label this. So this is going to be, I don't have a whole lot of space here, so I'm going to just use NT for neurotransmitter. Neurotransmitter receptor. So this would be kind of like at rest when there's not an active signal being sent across. But now let's draw this again of what's happening when this neuron is signaling the next one in line. So we'll recreate this wonderful artistic masterpiece and draw our presynaptic cell. And we'll draw our postsynaptic cell as well. And what's going to happen here, I'm going to do synaptic transmission in 4 steps. So the first step here, and let me use a different color for this. I'll use this nice pinkish color. The first step is going to be the action potential. Now, remember that action potential came down the whole axon, went to the axon terminals, and now that action potential got to the end of the axon terminal. So what happens is now we get to step 2, and that is the release of neurotransmitters. So our synaptic vesicles that are here that have neurotransmitters in them, those vesicles actually move to the end of the axon terminal. They fuse with the axon terminal end, the membrane, and then they release the neurotransmitters into the synaptic cleft. So now this synaptic cleft is filled with neurotransmitters, and that gets us to step 3, which is where the neurotransmitter binds to the receptor. And so let's go ahead and draw some of these receptors on the other side. But now what we're going to do is we're going to draw neurotransmitters sitting in those little binding sites on the receptors. And then step 4, what happens in the postsynaptic cell is a response. All right, so the postsynaptic cell can actually do different things depending on what neurotransmitter is being used. Let me explain a little bit better what I mean in this last space down here. So I'm going to draw another one. Draw another one of those graphs like over here. And we'll put time, and we'll put voltage. And we've got our minus 70 millivolt resting potential. And so this neuron is just chilling, it's resting at minus 70, and then it starts to receive some signals. Now, those signals can be a little bit different. One possibility is an excitatory neurotransmitter, and that will send the voltage going up. Another thing that could happen could be an inhibitory neurotransmitter. And an inhibitory neurotransmitter would send the voltage going down the other way. And what ends up happening is a neuron can be receiving kind of both. So let's imagine that this neuron is receiving inputs, and you get an excitatory input from here. And then you get an inhibitory input from here. So fire an action potential. Don't fire an action potential. No, don't fire an action potential. Fire an action potential. Fire an action potential. So now it's kind of like a vote. And we've got 3 votes to say fire an action potential and 2 votes that say don't fire an action potential. And as long as we reach kind of like a majority right here, then an action potential will fire. Oops, I drew that a little bit messed up there. And this majority right here is minus 55 millivolts, and that's called threshold potential. So in other words, a postsynaptic neuron won't just fire a response as soon as the first excitatory neurotransmitter comes in. Instead, a postsynaptic neuron counts up all the votes, figures out which one is the majority, and as long as there's a majority of excitatory neurotransmitters, then the postsynaptic neuron will fire an action potential. Wow, take a look at that. That's so much information about how neurons work. We've got an idea about the anatomy of one of these neuron cells, the 3 different parts, dendrites, cell body, and axon. We saw how a neuron signal is an electric signal of an action potential that involves the movement of ions, sodium and potassium, across the membrane differently. How that electric signal can go fast, non-myelinated axon, or faster, super fast, by having a myelinated axon with specialized Schwann cells that provide the myelin that allow the action potential to jump. Once that electric signal gets to the end of a neuron, now we switch to a chemical signal of synaptic transmission for the signal to jump from one neuron to the next. The electric signal getting to the end of the axon at the axon terminal causes release of neurotransmitters from the synaptic vesicles into the synaptic cleft. The neurotransmitters bind to neurotransmitter receptors on the postsynaptic neuron, and depending on the flavor of those neurotransmitters, we might get a different response. Having a majority of excitatory neurotransmitters causes the postsynaptic neuron to fire an action potential, but if things are skewed towards inhibitory neurotransmitters, then the postsynaptic neuron won't fire. This is a lot of information, and hopefully having it all in one place will help you understand how neurons work.